Three-dimensional tumor spheroids can mimic tissue-specific properties of tumors in vivo, providing more clinically relevant data for anti-cancer drug discovery than simple 2D cell culture. Our imaging technique, optical coherence tomography, can easily visualize the 3D structure of a single tumor spheroid with a size of several hundred microns and provide more accurate characterization of its morphology and drug effects, often within a few seconds. Using 3D spheroid model, we can potentially shorten the drug discovery timeline, reduce costs, and bring new medicine to patients more effectively.
Forming the spherical shape of tumor cluster is one critical step in our experiment. It is important to choose the right ultra-low attachment plate with the round bottom and the proper centrifugation speed after cell seeding.
Start this experiment by growing cells of interest in a culture flask as described in the manuscript. Maintain the cells in an incubator under standard conditions and monitor their health status every day. Refresh the media as needed. To perform 3D cell culture in multi-well plates, first remove the medium from the cell culture flask and wash the cells with sterilized 33 degrees Celsius pre-warmed PBS. Then add 1 milliliter of trypsin EDTA to resuspend the cells and incubate for 3 minutes at 37 degrees Celsius. Add 3 milliliters of culture medium to dilute the trypsin. Transfer this cell